Hello everyone. We are going to explain membrane attack complex. This is the table of contents. MAC is the final product of the activation of a protein called complement, which is responsible for puncturing the bacterial membrane and eliminating it through changes in osmotic pressure. They are formed on the surface of the cell membrane of pathogens by activation of the host complement system, especially near the C3 activation site, and act as a factor in the immune system to disrupt the lipid bilayer by leading to a transmembrane hole in the plasma membrane of target cells, causing them to lysate or die. Next, we will explain structure. MAC is composed of the component molecules C5b, C6, C7, C8, and C9, and many proteins are involved in its construction. The active C5b molecule binds to the C6 molecule to form the C5b6 complex, which then binds to the C7 molecule to form the C5b6-7 complex. The C5b6-7 complex combines with the C8 molecule to form the C5b6-7-8 complex. When C9 undergoes a conformational change, the hydrophobic region is exposed, and C9 enters the lipid bilayer of the target membrane. The altered C9 binds to another C9 molecule, which in turn binds to another C9 molecule, thus creating a chain reaction. The resulting C9 molecule forms a ring, forming a hole that penetrates the biological membrane. The complement has three pathways to activation. First, classical pathway. When the antigen binds to the antibody, it is bound by complement C1. Then, C1 is activated. Next, C4, C2, and C3 are activated in order. Second, alternative pathway. In this pathway, some bacteria directly bind to C3 but not antibodies and activate it. For that purpose, factor V or factor D in blood are needed. Third, lectin pathway. MBL, mannose binding lectin, produced in the liver, binds to bacteria and activates C4. Then C2 and C3 are activated in order. C3 activated by three pathways activates C5 and becomes MAC. What is function of MAC? MAC disrupts the lipid bilayer by leading to a transmembrane hole in the plasma membrane of the target cells, causing them to lysate or die. Thank you for listening.